What is secular humanism? With your host, Bernie Dehler. This is April 27th, 2011. Hello, everybody. My name is Bernie Dehler, and I'm the host today. And today I have a guest. He's calling in. It's Fazale Rana. He's got a PhD, and he's with the Old Earth Creationist Ministries called Reasons to Believe. Welcome to the show, Fuz.
Well, thank you for having me, Bernie.
Your full name is Fazale Rana, but you said your friends call you Fuz, so I'll just call you Fuz. How's that sound?
That sounds wonderful. I consider you a friend, so please call me Fuz.
That's good. And I consider you a friend also, and I'm also a former evangelical Christian, and so we might talk about some of our differences here too, and I think we can do this in a friendly way. So let's, well, let me show you first off some of the books that you have here, Fuz, and maybe you can talk about them a little bit. I'll bring up some of the screens here. So first we have, maybe just give like a 30-second to a minute description of what these books are. Here's your latest one, Creating Life in the Lab.
Yes, this particular book is about the attempts of scientists to create artificial life forms in the laboratory, single-celled entities that would begin to resemble life. And the book is essentially describing the work to date and the likelihood of scientists being successful in creating life in the lab, and then explores what would that mean to the Christian faith if indeed scientists are successful at creating life.
Yeah, so the idea is maybe when someday soon there's going to be a discovery of making life in the lab and Christians might get caught unawares, like how do we deal with this? And so you're trying to get in front of the curve to say this is how we deal with this?
That's exactly right, and I actually take a very positive perspective on the science and the technology associated with, again, the quest for artificial life. I think it's a great thing for both Christians and non-Christians alike. And so I'm trying to encourage Christians not to react with revulsion against the idea, but to think it through and realize that this is actually a good thing all the way around for both Christians and non-Christians alike.
Okay, and this other book is The Cell's Design, and I've seen that book, and I was really impressed with your knowledge. You know, you have a PhD in biochemistry, and I'm really kind of an armchair person. I mean, I'm just a casual studier on this stuff, so I don't know a tenth or a hundredth of what you know about this. So I was really impressed with your knowledge. So why don't you tell us about what this book is aiming to do?
Sure. Well, one of the reasons why I became a Christian was as a graduate student studying biochemistry, I became convinced that chemical systems inside the cell must be the product of a creator. And this book is essentially looking at what I believe to be the best evidence for design, if you will, in biochemical systems about 25 years after reaching that conviction again as a graduate student. And it's an attempt to look at, again, the different features of the cell and why I think they suggest the work of a creator. And the approach that I take in the book is kind of following in the footsteps of William Paley. It's using analogies to argue for design as opposed to other approaches that people have taken in more recent years, where the goal is to attack evolution. And I'm not really looking at doing that in this particular book, but rather trying to advance a positive case for design. Okay.
And then we'll talk more about some of the content in there, and maybe I'll challenge you with some things on that. This other one, Origins of Life, I don't know anything about this. Is this a new one?
This book is actually published in 2004, and it's a book that I wrote with an astronomer, Dr. Hugh Ross, where we look at the whole origin of life question and just simply talk about the inability of scientists to explain the origin of life, identifying what we believe to be, in many instances, intractable problems with evolutionary explanations for the origin of life. So this book is more a critique of chemical evolution than anything else.
Okay. Yeah, interesting. More questions to talk about that, so I'll make a note of that. The next one, Who was Adam?
Yeah, this book is looking at the question of human origins and essentially trying to make the case that the scientific evidence that's been uncovered in recent years is compatible with a biblical view of human origins in which humanity is derived from a single pair, an Adam and an Eve, if you will.
Okay, I know you have a pretty interesting chapter about pseudogenes. Is that the one where, is this the book that you had the most information about pseudogene arguments, or is this, or is there a newer one?
Both the cell design and Who Was Adam have a discussion of junk DNA, and probably the most extensive discussion is actually in Who Was Adam, but I believe the cell design is a little bit more up to date.
Okay. And then, okay, so that was it for your books. So now we'll get back to the whole point of this discussion. Let me give a little bit of background for our viewers. So you and Dennis Venema are two people with scientific degrees. Dennis Venema is an evangelical, you're an evangelical Christian. But you deny that humans evolved from animals. And Dennis Venema says, I'm a Christian, but I want to show you the evidence for evolution. And he believes it because of the evidence, he says. And then you guys had some back and forths, and it was starting to get into some of the details. And so now it seems like the discussion has been at least suspended, and so I wanted to pick up on some of the details. And the reason why I'm really interested in this is because this is, for me, I was an evangelical Christian, and this is a major reason why I left the faith. There's two reasons that we can probably talk about later, but accepting evolution, and then I was an evolutionist, evolutionary creationist, but then trying to make it work with theology, I found a lot of problems. And then there was another problem, too, I'll talk about later if we have time. So anyway, this evidence for evolution, I think, is really interesting for me. And I think it's something that the world is really starting to get a taste of now, because this all happened since the genome has been sequenced, relatively recently, the year 2000, 2003 or so. And all these other genomes, all the other DNA from these other creatures are being sequenced and compared. So I want to go over some of the details that Dennis Venema has in a YouTube presentation where he says this is the evidence for evolution, and I want to get your response on it. So what would you like to add to that?
Yeah, well, I think you're spot on in terms of, that the revolution in genomics that, is somewhat launched with the sequencing of the Human Genome Project, although there were genomes of other organisms sequenced before the human genome. But that revolution in genomics has really brought to the forefront the whole discussion about, the validity of biological evolution and the idea of common descent and common ancestry, particularly with respect to human beings. And so many evolutionary biologists are claiming that the so-called molecular fossils in the human genome and the genome of other organisms represents, in their mind, the best case for biological evolution. And so I think it's really, this is going to be the centerpiece of significant discussions among evangelicals in the next several years. Is there really evidence for evolution? And if so, how do you make sense of that within the context of the Christian faith? And I am skeptical about at least aspects of the theory of biological evolution. Not totally skeptical of all aspects of the theory, but some aspects I am skeptical, whereas people like Dennis Venema and others feel like the evidence for evolution is overwhelming. So it is going to be a centerpiece of significant discussion.
Okay. Now, some of this information is highly technical, and instead of explaining it too much, I think I'd rather just lose some of the audience, and there's going to be a certain amount of the audience who can pick this up, and then some people can watch it over and over again. So I think it's going to be really interesting that there may be a lot of people that want to see what your opinion on these things or how you explain things. And so, okay, let me go to the first slide. Okay, so this is taken from the YouTube Dennis Venema's presentation. So he has his classifications of homology, redundancy, syntheny, pseudogeny, and parsimony. And so I'm going to go through these kind of briefly, each one of these.
Sure.
And then get your take and redefinition if you want. So we go to the slide here where he shows the human, chimp, dog, cow, mouse, rat, chicken, and zebrafish. This is a small sequence for the gene that codes for insulin. It's the insulin gene is a hormone. Now this is, I blew this up actually from what his document is so we can see it halfway decently on the screen. So anyway, what you see here is that the human and the chimp, for example, so this gene here is for insulin. Every one of these animals has insulin. Now what you do is when you look at the human and the chimp, which according to evolution are closely related, you see that everywhere this lines up, this is just only the starting sequence of this too, but everywhere it lines up except for two in this excerpt I'm showing. And what happens is, this is not even down at the very DNA code level, this is the amino acid level, and you'll see that this A switches to a V, that's an amino acid, and this other one, A, changes to an S. So the argument here from homology is that when you look at the human and chimp, you see they're very similar, and then the more you look down, the more they start to diverge. And so that seems to indicate that there is some kind of descent because of that evidence. So that's the argument basically. So how would you like to rephrase that or restate it or refocus or anything?
Yeah, and so again, I understand, appreciate the nature of the argument, and the idea is that if there's common ancestry, as organisms begin to diverge and separate from each other and generate different lineages, those lineages are going to undergo independent evolutionary events that will alter sequences, and that you should be able to build evolutionary trees from similarities and differences in the sequences as a consequence of that, and that the further back in time the divergence happened, the greater the degree of difference that you expect to see. Or the more closely related in evolutionary terms, the more similar the sequence, the less closely related, the greater the difference. And, to me, homology and similarities in sequences like this are not necessarily problematic, because you would expect that a creator is going to utilize common templates and common designs when he creates. But at the same time, we live in a world where there are natural processes at work as well. And mutation is a reflection of those processes. And so you could easily look at these differences arising as a consequence of mutations, and you could also understand those differences to reflect the time of creation. So if an organism is created in the more distant past, you're going to expect it to accrue a greater degree of mutational differences compared to an organism created in the more recent time. So some of that difference could easily be accounted for, again, through mutation, but doesn't necessarily have to reflect time of divergence, but could be understood as time of creation. Also, we're making assumptions here that these single amino acid differences are non-functional, and that very well may not be a legitimate assumption to make. And if indeed they are functional differences, then that again makes sense from a creation perspective.
But no, wait a
second. You would argue that was intentionally introduced.
Well, on this one here, the function is the same as far as its insulin.
Right, but again, there's been work done, for example, that is showing that what are called synonymous mutations, which this is a mutation that's happening at the DNA level, well, all the mutations happen at the DNA level, but where the codon that specifies the amino acid is altered, but it's specifying the same amino acid. And yet it turns out that those synonymous mutations, which have historically been viewed as being neutral mutations, actually impact the rate of